Good afternoon. It's my pleasure to introduce today's Wednesday afternoon lecturer, Dr. Takjip Ha. TJ, as his friends call him, is currently the Guttel Professor in the Department of Physics and the Center for Biophysics and Computational Biology at the University of Illinois at Urbana-Champaign. He's also co-director of the Center for Physics of Living Cells at the same place. He grew up in Korea, in South Korea, has a bachelor's degree from Seoul National University, and then got his PhD in physics at Berkeley, and did a postdoctoral fellowship with Stephen Chu, who, as you may remember, was a recent former Secretary of Energy, and of course a Nobel laureate as well. After his postdoc, he took an academic position at the University of Illinois in the Department of Physics and the Center for Biophysics and Computational Biology, where he has been through his entire career studying single molecule dynamics and really has been one of the pioneers in what is one of the most exciting fields in biophysics and molecular biology. So as a graduate student, he pioneered the field of single-molecule Forster-resonance energy transfer, known as SMFRET, and went on to establish this technique as one of the most powerful ones for the study of molecular conformation dynamics of single molecules. He's made seminal contributions to the use of SMFRET as a powerful tool to directly observe and quantify biomolecular conformational dynamics. He's pioneered the development and dissemination of virtually every aspect of SMFRET, from instrumentation to measurement paradigms to improvements in fluorophore stability and labeling schemes, in addition to analysis methods and novel applications of SMFRET in single-molecule fluorescence approaches. More recently, he developed genetically encoded in vivo fluorescence-force sensors and combined these with immunoprecipitation at the single molecule level to develop a interesting new technique which he calls SiMPull, the SiMPull assay, and that itself has been a real advance in this field. I think the work itself, which involved lots of technological development of software and so on, has allowed us to gain insights into the details of genome maintenance processes, including SMFRET approaches to the structural dynamics of DNA, helicase function and activity, in addition to nucleic acid processing more generally. I would like to point out that Dr. Ha has also been awarded many prizes for this work, including the Ho-Am Prize, which is a Korean prize which is the equivalent in Korea of the Nobel Prize, and for which I think he deserves considerable credit. So I promised that he'd have at least 5 minutes to give his talk, which is entitled Genome Maintenance Up Close and Personal, Eavesdropping on Single Molecular Conversations. TJ.
Thank you very much. Well, I'd like to thank Dr. Gottesman for his very nice and also long introduction, so I don't have to speak for too long. And I'd like to thank colleagues here for inviting me. Should also thank the US Congress for keeping the government running this week, because my original trip was scheduled in this October last year, but I had to cancel because of the shutdown of the government. It's my great honor to be here. There are so many friends and colleagues at NIH I admire, and also really amazing and excellent students and postdocs that I had a chance to meet today or in the past. So DNA in a body undergoes insert all the time, and so you have problems caused by DNA damage. And if you're like me, look at the mirror in the morning, and I realize that I am getting older, and many other issues. So if you actually calculate how much DNA you make in your lifetime, actually you make about 1 light year of DNA. It's not the unit of time, it's the unit of length, how far the light travels in 1 year. That's mainly because DNA is very thin, also because we have a lot of cells in your body. So the cell has a daunting, or your body has a daunting task of keeping the DNA intact in the presence of a lot of damaging agents, and you have so much DNA to make and to keep intact. So there are many ways to do this, and many DNA repair mechanisms. And one topic that I want to mention briefly at the beginning is called homologous recombination. If DNA can have sex, this will be it, because DNA molecules of the same sequence can actually pair up to exchange strands with each other. And this happens first from damaged DNA that is broken in the middle, and then enzymes can make a single-stranded DNA that then searching for the matching sequence in another DNA inside a cell. And this process is mediated by a filament formed by proteins. In bacteria, it's called RAK, forming a filament around the single-strand DNA. And once you find the target sequence, then you can start homologous recombination and DNA repair. Of course, even in bacteria, you have millions of base pairs that are potential mates, and so it's actually quite difficult to find the right sequence in the sea of other competing sequences in the cell. So you can view it as a process of finding your soulmate. This may resonate with young postdocs here. So one way to achieve this is to find a random person on the street. So that would be like finding a soulmate via 3D search. So I have an animation that I made. Just date somebody randomly, and if it's not the right fit, then you come off and then try to find somebody else until you achieve the right match. But if you have 3 million different positions, it can be a very lengthy process, may not be very efficient. And okay, here I made it so that it finds a target, but you'll have to be very lucky to find that. So another possibility is called 1D sliding. And so the idea is that instead of dating a random person, you actually join a club of people who share common interests. You join, for example, a knitting club to meet like 20 or so people who like knitting, and then sample those people and locally. So that's one idea. And then if you do 1D sliding, then you can actually possibly accelerate the target recognition. So we use a method called single molecule FRED. The idea is actually quite simple. If you want to measure conformational changes of an enzyme, you can label the enzyme using 2 dye molecules, green and red, so that you can distinguish between the closed and open conformations. So in the closed conformation, 2 dyes are very close to each other. When you excite the green molecule, the energy is transferred to the red molecule, you get red emission. Whereas in the open conformation, you excite the green, you get mainly green signal. If you want to measure how quickly the butterfly flaps its wings in ensemble, you must synchronize the reaction by first convincing them to start from a single conformation. They start from one conformation. You can frighten the butterflies into taking off the leaf and perform a relaxation analysis. Well, oftentimes what you measure is not the wing flapping frequency, but how long it takes for the butterflies to take off the leaf. So what you want to do ideally is to look at a single enzyme and as a function of time so that you can observe anti-correlated fluctuations of the red and green intensities and deduce the frequency of the dynamics and also the amplitude of the motion. And this has been actually a really powerful method that is being used by many, many people in the world. So I have another slide here just to drive home the point of how it works. And if you have to Google Gangnam style, you will not enjoy this slide. So as the enzyme dances between different conformations, you measure changes in relative intensities. So actually, okay, so the audio is not on. So I'm showing you me dance with glow sticks of green and red color. Actually, that was not me. I'm here. It was in Aspen Opera House, and my son was dancing on the stage. I bribed him, telling him that, oh, I'll take you to private ski lessons if you dance on the stage for the general audience. So the idea here is that, unlike in the movie where you can see two glow sticks separately, you cannot do that in real biomolecules because they are too small. So what you are seeing is you're just seeing one spot, but different colors depending on the relative intensities. So you can go from yellow or to green and red or back and forth, okay? Finally until a reaction happens. So that is the idea. So you can follow the intensities of single biomolecules and then deduce the dynamics of the molecule during the function. So you can also use it to measure intermolecular interactions. When a protein moves on the DNA as a motor protein, you can actually measure the intensity change as a function of time. As the protein approaches the end of the DNA, then the green signal goes down, red signal goes up. And this animation actually is not quite realistic. The signal is a bit too clean, but our data is not very far. You can actually see real single molecule data showing these nicely anticoulomb changes where one is going down and the other is going up gradually. The surprise here was that when the enzyme goes to the end of the DNA, it somehow goes back to the beginning and repeat the process many, many times. And so based on many kinds of experiments, we were able to deduce what was going on and even propose biological implications for this cycle. So we published this paper in a journal. And then I paid an artist in Champaign-Urbana a lot of money to make a cover illustration for the magazine. So here the old man is reeling in the DNA and then kicking off the proteins bound to the DNA. But unfortunately, they didn't take the cover, so they instead used a picture of a diabetic mouse. So disappointing, but you lose some, you win some, it's fine. Here's another candidate we submitted that was rejected also. And my student, Jihae Park, is now acting as a superhero to kick the asteroids of the DNA. Anyway, so that's the kind of things we can do. So coming back to the target search by the Recce filament. So we're trying to see whether 1D sliding is occurring. So we labeled the DNA with one dye and then target DNA with another dye. And then we were able to see intensity changes of red and green between many different levels, consistent with the idea that the DNA filament can slide on the DNA, possibly in search of the target sequence. So this was published in a new open access journal about a year ago. And but this target DNA didn't have the matching sequence. So if you introduce a matching sequence, actually, if you introduce two partially matching sequences here and here, then what happens is that when this, at one position you get a high Fred, mainly in red signal, but then it realizes it's not quite a perfect match, and then it jumps off and then slides possibly, and then to land on the other position, and then the matching is not good, so it just hops back and forth between the two partially matching sequences. So it's, if I make the analogy again, you're dating twins, and then none of them is a really good match. So just go back and forth between the two. So, so that's the kind of measurement that we can take. And students in Columbia University, actually students of Eric Green, who used to be a postdoc here with Kiyoshi Mizuchi, made a movie about our data, and also another paper from California, and it's shown here. So we think that this can make the DNA repair up to 200 times faster, because filament can slide over that distance, about 200 base pairs, before falling off. It's a happy ending. So you can imagine 3D search and 1D sliding both contributing to the target recognition. You may have also noticed that there is another process mentioned, intersegmental transport. When the DNA and filament is long enough, you can actually maintain contact with the previous before, while sampling another sequence. So if I make that analogy again, you can stay in your current relation while sampling another person to find a better match. And that could also make the process much faster, according to Steve Kowalskowski at UC Davis. All right, so wanted to come back to the general theme of DNA repair. And during DNA repair, especially in replication, single-strand DNA is generated. And that has to be transient. Otherwise, actually, it's often a sign of trouble. In cancer cells, single-strand DNA can accumulate and so on. Many proteins bind to single-strand DNA. In bacteria, you have a protein called SSP, single-stranded DNA binding protein, and RECA, forming a filament. And helicases unwind DNA usually, but they can also move on the DNA as a motor protein, using ATP as a fuel molecule. In human cells, we have counterparts called RPA and Ref51 and some of the disease helicases. These proteins also talk to each other. One protein can recruit the assembly of the filament on the DNA, and that actually is done by gene implicated in breast cancer. And human disease helicases are known to also regulate the filament formation negatively. None of these proteins have strong sequence dependence. So the question is that how do they actually correlate or compete with each other, essentially working with the same common target DNA structure. So we extended our FRET technique to 3 colors to be able to measure more complex systems. So if you have a third color in purple, you may be able to gain additional information about a complex system. We also combine our single molecule FRET method with single molecule mechanical manipulation method called optical trap, where we can use the force to stretch the DNA, applying force to the DNA structure, and then measure conformational changes of the DNA using FRET as a function of applied force. So I want to use actually these technologies to look at the SSP protein on the DNA. SSP stands for, again, single-stranded DNA binding protein, forms a homotetramer, and its disordered tails are not seen in the crystal structure, but used to interact with many proteins functioning in DNA replication and repair. And as this cartoon shows, the SSP protein shown here binds to the lagging strand single-strand DNA And DNA actually wraps around the protein twice, shown in black line here, before it moves on to the next SSP tetramer and so on. And this has some physical basis because if you look at the crystal structure of the DNA wrapped around the protein core, you see about 60-some bases of DNA going around the protein, just like seam in a baseball or a tennis ball. And this tight wrapping is very nice because you can protect the DNA from degradation. And we found that once binding occurs, essentially it doesn't come off unless you have competing DNA or protein in solution. But this also raises an interesting problem. First, parking lot problem. If you have a protein with such a large binding site size, and if the protein binds to the DNA and it does not move around, then you can have one protein binding here, another protein binding next door, but you may have a gap of 20 bases of DNA that cannot be protected by the SSP protein. So that's a parallel parking problem. Another question is if the protein is so tightly bound to the DNA, and how do you remove the protein from the DNA when you must? So in this talk, I'm going to use this cartoon to illustrate the structure of the SSP as a tetramer. And the black line goes around the protein, representing DNA wrapped around the protein core. And you can imagine that if you put these dimolecules at the ends of the DNA, that should give you high FRET, mainly a red signal because of proximity. What happens if you have a DNA that is slightly longer than the binding site size? And this is what we thought would occur. It's called the rolling model, where one end of the DNA can detach from the protein core through thermal fluctuations. And most likely it'll go right back, but occasionally the other end of the DNA may capture that freed up protein surface. And this can continue as one-dimensional random walk. And if it goes on for some time, you may actually have a protein diffusion on the one-dimensional DNA lattice. And that actually will actually solve this parking lot problem. So we wanted to see whether this happens. So we labeled the DNA at 2 positions, so that in one extreme position of the protein on the DNA, you get small separation, you get high FRET, and when the protein rolls a little bit, you get an intermediate FRET value. At the other extreme position, you get the lowest FRET, and so on. So if this actually is happening, then you should see fluctuations of the intensities between many different levels on the single DNA protein complex. So this is the actual experiment. We have DNA about the binding site size wrapped around the protein, but we have additional 12 bases of DNA to give the protein additional room to move around. And then when we look at single DNA protein complex as a function of time, you see over about 40 seconds period, you see beautifully anti-correlated changes. If you calculate the efficiency of FRET as a function of time, it's not a two-state fluctuation. You see changes between many different levels, suggesting that it is indeed rolling between different positions on the DNA. One control I want to share with you here is the following. You can actually use a single-stranded DNA that is complementary to the 12 bases overhang to form a double-stranded DNA. Then you should lock the protein in one state where you have the highest FRET value. And so if the signal is coming from one-dimensional diffusion, then that should give you a stably high FRET. And indeed, that's what you see from a single DNA molecule. You see red signal staying high for all of the observation time. So based on this data and many other kinds of data, we believe that we have actually good evidence that we are directly observing protein diffusion on single-stranded DNA. As many of you know, proteins are already known to diffuse on double-stranded DNA to find the target sequence or damaged DNA and so on. But until this work, there was not actually a direct imaging, direct detection of protein diffusion on a single-stranded DNA or RNA. So we can do some math and then propose that step size of random walk is about 3 nucleotides of DNA, and stepping time is about 10 milliseconds at 37 degrees. So it can roll back and forth in that three at a time, about 100 times a second. One more control I want to show you here is a 3-color experiment. So you can label the protein with a green dye, and then label the 2 ends of the single-stranded DNA with red and purple dye molecules. And then you can measure Fred efficiencies to the red molecule and purple molecule shown here as a function of time, and show that these are actually also anti-correlated, meaning that when the protein approaches one dye, it is moving away from the other dye and so on. So ruling out the possibility that we are simply measuring the flapping of the DNA on the protein surface. It's actually moving physically on the DNA lattice. So this also solves a problem of how to remove SSP from the DNA when you have to. When Rk filaments forms and extends on the single-stranded DNA, it does it by adding one protein at a time to an existing end of the filament. You can imagine that protein rolling back and forth can free up space for additional binding of a Rk protein that prevents the protein from rolling back. And you're converting basically one-dimensional random walk into a directional motion to make the process much more efficient. So we have really interesting 3-color data that we published to support this idea. Another observation that we published is that if you have a hairpin structure in the middle of the DNA, then SSP protein diffusion can also melt the hairpin transiently. And then as a result, you can form a filament of the Rk DNA repair protein on DNA molecules that can form secondary structures. This can be like 30 times faster compared to formation in the absence of the protein that can transiently melt secondary structures. So the work was done by my former student Rahul Roy. Actually, this so he had a nice hit here. So he left my lab, and then another student joined the lab, Rubozo, and he told me that he actually doesn't believe the rolling model is the only possibility, and he proposed another possibility called the sliding model. And here's an animation to illustrate a version of that sliding model. So in this sliding model, defect can form on the DNA, but before, instead of actually going back, it can form a bubble, and this small bubble can then migrate back and forth, and if it annihilates at the other end of the DNA, then protein would have migrated by the side of the bubble and taking a step. So that actually will be also fully consistent with our published data. So my student then proposed that we do one experiment among many shown here. So if you now put a dye on the protein and then dye on the middle of the DNA, then in the rolling model, all of the action is going on at the ends of the DNA. So there should be no change in the signal between the two. Whereas in the sliding model, two molecules are moving all the time, and that should give you fluctuations in Fred that we measure. So he did that measurement, and then he showed that you see Fred fluctuations of similar time scale and magnitude as before. If you do a control where DNA is too short for migration to occur, then you get a steady signal within noise. So based on this and some other kinds of data, we actually now prefer the sliding model over rolling model. By how much? By this much. Another interesting question that we can address is whether SSP diffusion requires the fully wrapped structure that I discussed, like as in a baseball or tennis ball seam. And if the answer is no, it'll be more interesting because human proteins, RPA and so on, are not known to have these fully wrapped structures. So the strategy here is to use, optical tweezers. If you want, you can call it chopsticks are made of light, to grab a bead to pull on the DNA, to apply force to the DNA ends of the SSP-bound single-stranded DNA, and measure Fred to determine what forces are needed to unravel the DNA from the protein surface to prevent the fully wrapped structure. And then does diffusion still occur at these forces? So to give you the answers first, we need fairly low forces, 3 to 5 piconewtons. These are actually quite low forces. And we still see diffusion from these forces. So let me just show you some data obtained using the instruments shown here. So you're measuring basically Fred as a function of time, where as you increase the force, so the example is shown here, as a function of time, you can increase the force shown in gray curve, and then at high force, you come back to the original value and then do it 5 times, and measure the intensities of the red and green molecules on the DNA. And you can see really nicely anti-correlated changes. And you can also calculate the Fred efficiency that decreases gradually with force. So when my students showed me the data first time, I almost cried because the data was so beautiful. But I didn't, and as all good advisors do, I told him to go back to the lab, take some more data. And so he did, and so he was able to average many molecules like this, and show that Fred drops with force, and by the time you go to 4 piconewtons, so basically DNA has been fully unraveled from the protein surface, and you don't have the fully wrapped structure anymore. The question is then does the protein diffuse under these conditions? And so the answer was yes. So we think that this is quite general to many other proteins. In fact, now there are some data in the community showing that human proteins also diffuse on the DNA, and evidence produced by the same kind of assay we developed. Finally, there are 2 actually ways to slide. One is this sliding with bulge model or reptation that I described to you earlier. But technically, it's possible that the DNA could dissociate from the protein entirely, and then protein rotates a little bit and then rebind, so sliding without the bulge model. And the prediction, basically based on our expectation, was that if you apply force, then you prefer this unraveled state. So in this model, then that should assist the diffusion of the protein. Whereas in this model, when you apply force, you can interfere with the formation of the bubble, which is required intermediate, and that it should resist diffusion if you apply the force. So we measured how quickly diffusion occurs as a function of force for 2 different solution conditions. In both cases, diffusion became slower with force, and this actually supports the reptation model, sliding with bulge. So this is like what we proposed a couple of years ago based on these types of experiments. Now, Fred is sensitive to DNA molecules up to about 100 nucleotides in length because it's blind to distances beyond about 80, 90 angstroms. But in the cell, DNA can be much longer. Single-cell DNA can be much longer, thousands of bases long. So we developed, actually my student developed, Kyung-Suk Lee, another way to stretch the single-cell DNA sideways using optical trap, and then use what's called tough microscopy to image the proteins bound to the DNA directly. And first, he used it to look at a helicase called EBRD helicase, where Yang is really well known for that protein in NIH. And this protein is known to use ATP to move from one end of the DNA, 3' to the 5' end of the DNA. And so this is where the bead is attached, and this is where surface tethering is done. And let me see if the movie works here. There's no music, don't worry. So you can see something appearing in the middle of the movie, and it's going down gradually along the length of the DNA directionally on the single-strand DNA. And you can also notice that it gets brighter and brighter. So why? Is it collecting cosmic dust? No. That's because in turf, your excitation decays exponentially away from the surface. So as the protein approaches the surface, then intensity goes up. So you can plot what's called a Keema Graph, So plotting the position of the protein as a function of time, you see really highly uniform motion of this protein. And in fact, in some lucky cases, we can see many molecules binding and moving down the DNA in succession on the same DNA molecule. They all show the same slope that actually you can use to determine the speed, fully consistent with ensemble data by Tim Norman. And I call this my meteor shower because it looks like meteors falling on the night sky. So I can watch this image for a long time. So then we decided to look at SSP directly. Does it actually move when you look at this protein under the microscope? And the answer is yes. Okay. So here's another movie. You can see actually two of the molecules, and then they both actually move up and down, suggesting a one-dimensional random walk. And you can plot the Keema graphs for several examples here. They will show long-range diffusion on the single-strand DNA. So we are very happy about the data until we actually did some numbers. So you can actually look at how diffusion occurs and calculate what's called the diffusion coefficient. And we got numbers that are very large. These are 100,000 nucleotides square per second. I know it's a weird unit, but in our earlier paper, we proposed that this number is actually 1,000 times smaller. And that was done on short DNA. So we actually think that this is both correct. And when you go to long DNA, you have some new things that happen that you don't see on short DNA. And so the idea is that because SSP has multiple binding sites for the DNA, then on the force, it may be unraveled a little bit, and then it can then grab DNA at a distant location to form this type of loop, and then jump over to another segment. And this can make the process actually quite fast. Intersegmental transport I described to you earlier, or direct transfer. Prediction here is that when you apply force, then it'll become more difficult to jump long distances because DNA becomes straightened up. And in fact, when you measure the diffusion coefficient as a function of force, you see reduction in values, and we can also come up with a toy model to account for the data to a certain degree. So it is an example where when you change the length scale of observation, you can start to see some new things that you didn't see before. Okay, so mainly I talked about this SSP protein diffusion on single-strand DNA today, using single molecule fluorescence and optical tweezers, fairly sophisticated in vitro technology applied to purified or synthetic biomolecules. The next part, I want to spend just maybe 10 minutes to tell you about a different direction that we are pursuing called single molecule pull down. And actually, if you're falling asleep earlier, you can just wake up and listen to this because it has no relation to the previous part. So now 6 years ago, I went to Keystone Meeting. I went there actually mainly to ski, but I was also interested in learning about innate immunity and signaling mechanisms. So I went there and just to attend the meeting. It was very interesting that actually I didn't know anybody there. I didn't even give a poster, and some people were asking me whose lab I'm working in. So I was so happy. I felt young again. And so Kiyoshi, you should try that. Yeah. Just kidding. Actually, someone was giving a talk, and then mentioned this LPS. You know, if you buy LPS from Sigma, it's no good. And I had no idea what LPS was. So I asked a student next to me about LPS, and she looked at me like, you know, I'm from Mars. So I know what LPS is now. Anyway, so I learned a lot of biology there, and these are probably topics for another talk. But one thing I learned is the fact that people use pull-downs and Western blots a lot. And because I'm a physicist by training, it was shocking to see talks with slides after slides of Western blots. And it took me a couple of days to learn how to read the slides. And then I realized that this is such a powerful tool. You know, what can be more important than measuring protein interactions in living cells, right? So I felt then very small and depressed. You know, if I were to give a talk at this meeting about little proteins moving on little pieces of DNA, no one would care, right? So what I did was to ski some more, and then came back. And I had a new grad student, Uncle Jane, and told him that, well, you know, why don't you try a single molecule Western blot? And if you succeed, then perhaps biologists will like us. So he, as all new grad students, he was clueless as how difficult things are. Anyway, so he called this single molecule pull down or SiMPull. So the idea is that, you know, in core immunoprecipitation that is carried out in many biology labs every day, use lyse a cell, and then you use a column with beads linked with antibodies against the protein of your interest or antigen of your interest, and then cell lysate contain, you know, protein complexes that contain that antigen. You can capture them, and then you can rinse away free other proteins from the column, and then collect what's left, and then, you know, you can run a gel, and you can do Western blots, or you can do mass spec to see what you have, how many copies you have, and things like that. So this is really a gold standard for measuring protein-protein interactions in living cells. There are some limitations. First, you know, if you use protein A to pull down protein B and C, in regular Western blot, you cannot tell whether what you have is the case where you have AB forming a complex and AC forming another complex, subcomplexes, or you may have all of them in one complex. Okay? Whereas if you do single molecule measurements, in principle, you can distinguish between the two possibilities. Another example is the following. So, if you do a really careful job, you may be able to determine the ratio between different components, assuming there is a single complex stoichiometry. But conventional Western blot does not tell you how many copies of one protein are found in one complex. Okay? So that's the stoichiometry issue. But in principle, if you do single molecule measurements, perhaps you can tell the difference. So this is how we do single molecule pull down. So we have a slide for the microscopy imaging. We passivate it using polyethylene glycol pack, and we use biotin to immobilize avidin molecules, and then biotinylated antibodies against the protein of interest. And then we add the cell lysate of appropriate dilution so that you can capture proteins in the concentration that is consistent, compatible with single molecule imaging. And you wait until protein complexes are captured, and then you basically wash away everything else, and then you turn on the laser and you detect what's bound to the surface. Okay, so if you have fluorescent fusion proteins, then you can detect them, and using again turf imaging, and they light up. It's pretty easy to do these days. Or you can also use labeled antibodies to do essentially immunofluorescence detection of the protein complexes. So we first used purified yellow fluorescence protein YP to validate the technology. So we can have anti-histidine GFP, anti-GFP antibodies, histag antibodies on the surface, and then our YAP is expressed with a histidine tag, and then you can capture them. So if you add 250 picomolar of YAP on a surface with histidine antibodies, you can see single fluorescent spots due to single YAP molecules. As a negative control, you can omit the antibodies and no proteins whatsoever. And then you still see some fluorescent spots due to the background fluorescence. And if you do control no antibodies but 10 times higher proteins, then you see the same level of background as the blank. And if you use wrong antibodies against a flag tag, then you also see a little additional binding. You can do many, many negative controls. You can even quantify the results by counting the number of spots per imaging area and plot them and show that, well, if you have the right antibody, then you get 600 spots per imaging area, but for all the negative controls, you get less than 50. So you can show that this is highly specific and so on. But that was for the purified proteins. But does it also work with the crude cell lysase? That is a big question because it's possible that cell may have lots of things that can overpower the binding capacity of the chip, or you may have also a lot of fluorescent impurities in the cell extract. But it does work, so you can do the same, but now with cell extracts expressing these YP proteins. These are all negative controls, and you can quantify them. In addition, you can also look at a single spot and measure the intensity as a function of time and show that intensity drops to the background level in a single step, consistent with the monomeric YP that we're expressing. And in fact, here you can show that histograms of spots showing 1, 2, 3, 4 photobleaching steps. It shows mainly single-step photobleaching, as you expect. If you instead express tandem dimer of YAP, then you start to see many molecules showing two-step photobleaching, showing that you have two copies. And a majority shows two steps, some showing one step, and very few showing 3 or 4 steps. And the reason why you do see one-step photobleaching is because not all of the YP molecules are active fluorescently. So if you assume that about 80% of them are active, then you can explain the data with the dimeric stoichiometry. So we showed that this works for crude lysase, and you can get stoichiometric information. So finally, we then wanted to validate this using real protein complexes. So we turned to our collaborator, Kevin Xiang. And protein kinase A is a famous kinase that functions downstream of GPCRs. And it forms an inactive hetero tetramer, 2 copies of regulatory and 2 copies of catalytic units. So, and it's known that if you add cyclic AMP, then the complex falls apart to liberate the kinase unit for downstream signaling. So Kevin's lab engineered R unit with a flag tag and M cherry red fluorescence protein, and C unit with HA tag and YP, and then showed that modifications did not perturb the protein's ability to form tetramer and also to dissociate when cyclic AMP is added. And it's shown actually here. This is the regular Western blot that says that fusion constructs work fine, and if you express just one protein at a time, and then you can do again the regular pull-down experiments, whether you use HA antibodies or GAP antibodies with negative controls. And then we co-express both proteins in the same cell culture, and we captured these complexes using an antibody against the R unit. So you should see red as fluorescent spots as shown here. But if the complex is pulled down, you should also see the yellow spots, and as actually in this case, green color here, YP spots are also shown, and we see similar copy numbers agreeing with the expectation. In fact, many of the spots co-localize, showing yellow or orange colors, and that we have real complexes. And there are many reasons why you don't see all of them showing a yellow color. We can discuss if you want. As an additional control, we can also add cyclic MP to the slide chamber. And the expectation is that the complex falls apart. You lose the green spots due to YFP, but you keep the red ones because they are bound to the antibodies directly. And indeed, when you look at the slides after such a treatment, actually you keep the red, but you lose a lot of the green. And if you look at the overlay, you have very few of them showing yellow colors, showing that indeed things fall apart as you expect. In addition, you can also do the stoichiometry analysis. If you express just one protein and pull down and then count the photobleaching steps, you see mainly one-step photobleaching. But if you co-express them, you pull down the red one and you can detect the yellow one as a function of time, and you see a lot of two-step photobleaching, and with actually some also similar number of one-step photobleaching, but very few as 3 or 4 was consistent with the expected dimeric stoichiometry. So based on these data, we think that the method actually is ready for wide application. In fact, we have helped many other labs work on their systems in various collaborations here. So I want to show you one slide about what the applications we have done. So we show that you can do this with membrane proteins and cytosolic proteins. We have some nice data on a protein complex called mTOR, for example, and yeast nuclear proteins, and mitochondrial membrane proteins, and even animal tissues. And you can actually get brain extract. The tube is pink because of the blood. The students are brave enough to put them into this single molecule imaging chamber, and they can actually do immunofluorescence to detect protein complexes. So essentially, almost everything we tried worked. And usually, when it fails, we failed actually in transfection and so on. So I think there are many things actually one can imagine doing using this approach. Let me stop here. I just want to acknowledge the hero of this project, Anko Jain, who is now a postdoc with Ron Vail at UCSF, and Kevin Shang's lab was really instrumental in this project, and Jie Chen is a collaborator on the mTOR project. Thank you very much. So that was 5 minutes, right? Yeah.
Hi. I did have a quick question for you on the photo bleaching. So I noticed you had some YFP monomeric controls with some two-step processes, and then some YFP dimeric controls with 3 and 4. What do you attribute those 2 steps for the monomer and the 3 and 4 steps for the dimer to?
So that is a good point. So when you see 2 steps in like 5% of the molecules spots, when you have monomeric constant, there are many different possibilities that we cannot really distinguish among. One is that even monomeric YP may still have residual activity to form dimers at low level, and we may also have accidental co-localization of 2 molecules that are independent of each other. Okay? And so these are the possibilities. So it's our ability to determine stoichiometry actually depends on these basal level signal, right? So if you find a way to reduce that further, we'll be able to make stronger statements about the stoichiometry. So we are focusing on things that we can say very clearly given the basal level of these kind of signals, but I think it can be done better.
Do you think that you could adapt the SiMPull approach for live cell analysis, for example, with nanobodies that will be expressed in the cell, localized to some component? Have you thought about that?
So I think maybe your question is not so much about whether we can do SiMPull for live cells. What I showed you is that you have to kill the cell to get them. But maybe you're asking the question of whether you can do
complexes.
You can determine the stoichiometry of the complexes at the single molecule level in live cells. The answer is yes. Actually, that was done actually many years before by Udi Isakov at Berkeley. So they expressed ion channels with fusion proteins, and then they were able to detect single complexes in living cells and counting photobleaching steps and determine the stoichiometry. That approach actually is good, for really tight complexes where even when you express them at low level, low enough so you can see single molecules in cells, they still form complexes. But in many cases, the concentration in the cell is too high to detect single spots, right? So here you can keep more or less, hopefully you can keep the complexes intact and then quickly lysing them and then diluting these complexes, you can start single molecule imaging very quickly. So that's the motivation here.
When you're looking at Rec A sliding on a single-stranded DNA, could that also be going by reptation, or could you tell the difference?
The sliding of Rec A filament?
Yes.
It is a good possibility. Reptation on a larger scale is, you can imagine, I showed my PowerPoint movie where the filament, rigid rod of filament is sliding as a rod on the DNA, which is also depicted as a rod. But it's possible that they may actually have some defects, like bubbles coming up like this, and that can also influence the kinetics. You may be able to jump over some distances there too. If you actually extend it to even longer lengths scales, like thousands of base pairs, then of course what you get is the intersegmental transfer that Steve Kwiatkowski proposed, where looping of DNA that is possible, on long length scales can also allow the protein filament to jump from one end to another end very quickly without fully dissociating from the DNA.
So let's thank Dr. Ha for a scintillating presentation. And let me invite you all to a reception in the library, and there'll be opportunities for more discussion there.